Good evening and welcome to our first webinar of 2025. We are so excited to kick off the new year with another session in the Immune Deficiency Foundation's Decoding PI series. Tonight's featured topic is gene therapy and gene editing, led by Susan Prockop of Boston Children's. My name is Emma Mertens, and I'm the Program Manager for Education at the Immune Deficiency Foundation. On behalf of IDF, we thank you for tuning in to this virtual event. Throughout this series, guest presenters will provide a deep dive into clinically advanced topics from the world of PI. If time permits, the session will be followed by an audience Q&A with our presenter. We hope through your participation in these programs that we may move forward in our mission to engage and empower the PI community through education. During portions of tonight's talk, we will offer audience polls. When a question pops up on your screen, you may submit a response by clicking one of the presented options. Only you can see your answer. Also, a reminder that closed captioning is available for tonight's programming. A brief disclaimer. Please remember the information presented during this meeting is not intended to be a substitute for medical advice, diagnosis, or treatment. We are here today as a trusted source and friend to provide you with information. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based on information presented during an educational event. This webinar is made possible by our wonderful sponsors. It is due to their partnership and contributions that we can provide programs like this for the PI community. Please join me in thanking today's sponsors. And now I'm so pleased to introduce our presenter for this evening. Susan Prockop is a notable pediatric hematologist-oncologist at Boston Children's Hospital and Dana-Farber Cancer Institute. She specializes in stem cell transplantation and cellular therapy, with particular emphasis on treating children with blood cancers and disorders. Additionally, she is an associate professor of pediatrics at Harvard Medical School. Welcome, Dr. Prockop, and thank you so much for joining us.
Thank you so much, Emma. It's a pleasure to be here. I will go ahead and share my screen.
Sure, and while you're doing that, I'm going to go ahead and launch our first poll. So if everyone can take a moment to answer that question, I'm going to leave it up for 30 seconds, and then we'll go ahead and see everyone's answers.
Can you see my screen okay?
I sure can.
Great. So as I said, it's a real pleasure to be here. And I hope tonight to give a little bit of history around gene therapy, especially gene therapy for primary immune deficiency, inborn errors of immunity, as well as sort of a sense of where the field is now and hopefully where we're moving quite quickly. These are my disclosures. None are specifically relevant to tonight's presentation. So, I thought I'd start with a little bit of an overview of the history of gene therapy. And really, this field has been most marked in the arena of treatment of inborn errors of immunity and started in the treatment of ADA deficient SCID in 1990. And in the next almost 35 years, as you can see here, there have been successes, but also setbacks with after the first gene therapy for X-linked SCID and Wiskott, the recognition that these therapies had the potential to have genotoxic effects. And then over the last 35 years, we have really developed safer tools with which to embark on gene therapy, including gene editing. So both gene addition strategies that integrate genes sort of randomly into the genome, as well as more specific targeted approaches to get a gene or just a mutation to a gene exactly where you want it to be. And now there are multiple approved products across hematology, immune deficiency, as well as retinal disease, and metabolic disorders. And so when we think about curative therapy for inborn errors of immunity, we have to recognize that there are over 440 monogenic disorders associated with inborn errors of immunity. And while based on the fact that these are part of the immune system and they are therefore potentially curable with hematopoietic stem cell transplant, some patients can be predicted to have inferior outcomes from transplant and should be considered for alternative curative therapies, including gene addition and gene editing. And I think really how I think about it when talking to a patient and their family and thinking about whether curative therapy is appropriate for them, you need to think about really what the need is, what it's going to do to alter their life, either their lifespan, their quality of life, their freedom from infection or functionality, as well as what the potential options are for both hematopoietic transplant and the outcome predicted with hematopoietic transplant. And then thinking about whether some of these novel therapies that are really, in some instances, in their infancy and in some instances more established is. So I think it's helpful to sort of think about what the pluses and minuses and how gene therapy is different from hematopoietic transplant as we start to think about where it can be added to the armamentarium of curative therapy. And so with gene correction or gene addition therapy that is stem cell based, and so far all of that therapy has been ex vivo, meaning taking the stem cells out, manipulating them, and putting them back in. We know from allogeneic, from stem cell transplant, what level of correction of the disorder is needed to effect cure. And there are some disorders where you need to have 100% donor cells across all lineages to be curative. And those disorders, at least up until now, are not really amenable to gene therapy. But disorders where you need only to have complete correction in the T cell population, or where you only need 10 or 15% correction to achieve sort of normal function, those are disorders that are amenable. So knowing what level of correction you're aiming for is critical. And then what level of chemotherapy or conditioning you might need to be able to get those new cells, those manipulated cells in. The potential for genotoxicity that we'll talk a little more about through this evening, then non-genotoxic side effects. And what does it mean that there's going to be some residual hematopoietic cells that are not genetically modified, at least with all of our current platforms. Alternatively, in the allogeneic stem cell transplant setting, there's a risk of graft versus host disease, of graft rejection. There's a potentially quite prolonged period of immune compromise and risk of infection that can be even more significant than the immune compromise that a patient comes into therapy with. And then there are non-genotoxic side effects from the conditioning and chemotherapy and the risk for treatment-related mortality. And here too, we do need to know what level of correction we're aiming for, and that helps us understand what level of conditioning we need, what type of graft we need. And so pivoting and thinking specifically about gene therapy for rare disease, we want these therapies to be safe and available and effective. And I think a lot about how we develop them, assess them, and use them in a setting of rare disease, such as inborn errors of immunity. And, some of the advantages of this type of platform are that it's a hematopoietic disorder. So we can do ex vivo manipulation of cells, put them back in and correct the defect. We know from transplant what level of correction we need. And there's no benefit of alloreactivity or graft versus host disease in this type of disorder. And there we have established centers of excellence. And at least in the setting of SCID, newborn screening for some of these disorders has really helped funnel patients appropriately to centers of excellence. The challenges are that these are rare. There are unique referral patterns for the different disorders that may or may not align with where the expertise of gene therapy is. There's variability in terms of disease manifestation, so making decisions about treatment can be very complicated. And gene therapy and gene editing are high-cost initiatives and high cost relative to patient volume. So we really need to think about operationalizing therapies that are maybe only going to ever be appropriate for a couple hundred patients at most for some of these disorders. And then again, thinking about the risk of genotoxicity, because SCID is really the first place where genotoxicity was identified in the setting of gene therapy. So just an overview of what we're talking about when we're talking about ex vivo gene therapy. So the idea is that hematopoietic stem cells are collected from the blood or bone marrow of the patient. The patient cells are then isolated. If you're going to manipulate the stem cells, you isolate the stem cells. The T cells, you isolate the T cells. You place into those, you put into those cells a gene, a corrected gene that is packaged into a delivery system that's usually a viral-based delivery system, or you can do something called electroporation, which is sort of making tiny holes in the cell and putting the gene in that way. And that the gene or the genetic material that you're putting in is integrated into the genetic material in the cell. And then these genetically modified cells, and because the gene is integrated into the gene of each cell is genetically modified, or as many ones as the gene got into, and those cells are then returned to the patient. And for most of the disorders we're talking about, we've really learned that prior to infusing the cells back into the patient, giving some amount of chemotherapy helps make sure that the stem cells engraft. And, you know, I've said a couple of times that it matters what level of correction you need, whether gene addition therapy or gene editing is appropriate. And immune deficiencies are sort of an ideal place to think about this, because we do know from transplant experience that for most of these disorders, we don't need to replace all of the hematopoietic stem cells. If you have low level of engraftment of hematopoietic stem cells, for instance, in SCID, you can get a normal number of normal T cells and a normal number of normal B cells, and you will have some low level contribution to all of the hematopoietic cells from the corrected hematopoietic cells that represent a small proportion of the total stem cell pool. Alternatively, there's some disorders where we know we need a very high level of chimerism, and that the risk of rejection remains quite high if we don't achieve that high level of chimerism. And so understanding the disorders that are amenable to low level chimerism and that get fully corrected with low level chimerism versus disorders where having abnormal cells still in the body can still create disease is very important. When we think about SCID, there are multiple potential targets of gene therapy in the context of SCID. And the ones that have already been quite significantly explored are ADA deficiency, X-linked or gamma chain deficiency, and Artemis. But I will talk about these more, but there are others all along these pathways, such as RAG deficiency, that are certainly being explored in terms of potential gene therapy. I mentioned genotoxic therapy, but there are other toxicities that we think about when we think about gene therapy. So genotoxicity is the idea that that piece of gene that you've introduced into a cell gets introduced in a dangerous place, in a place that then makes some other part of the cell malfunction or overfunction. And we think of the most dangerous sites as sites of proto-oncogenes, or sites where there's a gene that can push a cell to becoming a cancer cell. And so that's one form of genotoxicity. Another is that you disrupt an essential gene and make that essential gene stop working correctly. That can also be genotoxicity. The non-genotoxic components of this therapy include the risks with conditioning. So for many of the therapies I'm going to talk about tonight, we've come to understand that some level of busulfan, usually conditioning, is needed to get stem cell engraftment to get the stem cells that have been manipulated outside the body to engraft and stay in the person's body long term, in the patient's body long term. And so the toxicity associated with busulfan can be significant. And while the cells that have been genetically manipulated are not exposed to the busulfan, other elements of the hematopoietic system are exposed to busulfan. And then on target toxicity, what I mean here is that if the gene you've manipulated and put back in is over or under expressed, that is a dysfunction that's specific to the gene you're talking about. And then off-target toxicity is when you over or under express a gene at the integration site, similar to the genotoxicity that I first described. So early gene therapy and really gene therapy started with gene therapy for rare disease, was associated with genotoxicity. And these were all gamma retroviral based gene therapy vectors, and they preferentially integrated into proto-oncogene sites. And so with the first Wiskott-Aldrich gene therapy approaches, while 9 of 10 patients experienced major improvements in their immune system and platelet defects, the insertional oncogenesis resulted in 7 of these 9 participants developing leukemia. In the first trials for X-linked SCID, there were 20 infants treated with good immune reconstitution, but insertional mutagenesis occurred in 5 at the time of this publication, in 5 of those infants, and continues to be a risk for these infants. In the setting of chronic granulomatous disease, again, a first-generation gamma retroviral-based therapy in 5 infants, 3 of whom developed MDS. And so since that time, there have been 2 major advances in the way that we deliver gene addition, hematopoietic stem cell directed gene addition therapy. So the first is that even with those gamma retroviruses, if we deleted part of the elements of the vector that push high level expression of the gene, those vectors have been demonstrated to be safer, and the incidence of leukemia is much lower thus far in patients who've received therapy with those. And the second is really more ground shifting, is transition to lentiviral-based vectors. So lentiviral-based vectors are based on the HIV virus. They are very good at getting into hematopoietic stem cells. And unlike gamma retroviruses, they tend to integrate not right at the start site of a gene. They integrate into more benign parts of the genome. And so you also get, because they get into the hematopoietic stem cell better, you get more stem cells that are modified. And so you have a more polyclonal reconstitution. You don't have just one stem cell trying to provide everything for somebody. And so there are 2 settings that we think about hematopoietic stem cell-based gene therapy for in the setting of primary immune deficiency. And I tend to divide them into those where there are certain patients, especially those who don't have an option for a matched sibling donor transplant, where the non-matched sibling donor transplant are associated with inferior outcomes. These are sort of a very obvious target for alternative therapies. Now, unfortunately, in this setting of rare disease, the patient numbers are small, so it becomes very hard to truly compare one therapy to another. And the other part of it that makes it hard is that we can compare the short-term toxicities quite easily, but the long-term toxicities that may not appear for years, including the risk of genotoxicity, can be very hard to compare in a real-time way. And then both of these therapies are evolving. Transplant therapies are evolving. We have new contemporary approaches that are making alternative donor transplant safer. But gene therapies are also very rapidly evolving. And I'll touch on some of the newer things we're doing there. And transplant has certain risks that are completely avoided with gene therapy, such as the risk of rejection and GVHD. But in the gene therapy side, it is hard to assess the safety quickly. So we know not as much in the primary immune deficiency setting, but in other settings where leukemia has emerged after stem cell-based gene therapy, some of those patients develop the leukemia as late as 7 years after treatment. And so it becomes very hard to know when is it safe to say, I know what the incidence is. So I'm starting here with some of the immune deficiencies where we have demonstrated that there is inferior outcome after hematopoietic transplant if you don't have a matched sibling donor. And so this is ADA deficient SCID. And the PIDTC did a large retrospective study with very clear demonstration that matched sibling donor transplants and other matched unrelated donor transplants also are associated with very good disease overall and event-free survival, but that if you don't have a matched donor, the umbilical cord blood donors, unrelated donors, and mismatched related donors are all associated with inferior overall survival and event-free survival. And this is a place where if you look at overall results of patients who went to gene therapy compared to all patients who went to hematopoietic transplant, including those who had matched sibling donor transplant, there's no significant difference in the outcomes. And so that becomes quite compelling to think about gene therapy for ADA deficient SCID, even maybe in the upfront setting. And on the right-hand side, Don Cohn's studies of ADA gene therapy for ADA deficient SCID show very, very high overall and disease-free survival. In these 50 patients, there are only 2 patients who didn't have durable correction of their ADA deficient SCID. And in more than 50 patients treated worldwide with gene therapy for ADA deficient SCID, there's really been very limited vector-related events, even though many of these patients, so of the 50 patients, those are all patients who got lentiviral-based therapy. But worldwide, over 20 patients have gotten gamma retroviral-based gene therapy, and they're really very limited genotoxic events. There is, however, one that was only recently identified, and this is again, so gamma retroviral gene therapy for ADA deficient SCID has been employed for almost 20 years, and the first patient who developed T-cell ALL 56 months after treatment was just identified last year. This patient had integration of the corrective ADA gene into the LMO2 proto-oncogene site, which is a very common site of integration. That site of integration was detected for years and detected in multiple lineages, so it was clearly in the stem cell to begin with. But the T-cell blasts had additional mutations in addition, and you can see in the right-hand panel B that you have a lot of these are all T cells with a lot of different integrations. What's being shown on the y-axis is increasing number of integration, increasing diversity of integrations. So all these patients had a lot of integration. And then in the red, this patient who developed leukemia had a really sudden drop in the number of integrations, such that at some point, all of their T cells were coming from this one T cell progenitor that had an LMO2 proto-oncogene integration site and developed into leukemia. So and this is really about sort of ex vivo gamma retrovirus-based gene therapy. So in 2016, the EU approved GlaxoSmithKline treatment with gamma retroviral-based gene therapy for ADA deficient SCID, and it's called Strimbelis. It was the first ex vivo stem cell gene therapy to receive approval anywhere in the world. In the US, and this is sort of really based on a lot of preference for using lentiviral-based therapies, Don Cohen developed a lentiviral-based ADA gene therapy. And the saga here is that in 2016, a pharmaceutical licensed it, but in 2022, they stopped developing it and returned it to UCLA. So there's now an open trial with the goal of a commercial product. And so this open trial is for autologous gene therapy with a lentiviral-based vector for ADA deficient SCID. And it's open and enrolling. Another form of SCID where there are inferior outcomes, especially with alternative donor transplant, is Artemis deficient SCID. So Artemis deficient SCID results in mutations from the DELREC gene and plays a crucial role in repairing DNA breaks. And so when this gene is not working properly, DNA breaks aren't repaired properly. And so this results in sensitivity to radiation, sensitivity really to anything that breaks the DNA, and sensitivity to alkylating agents. So these infants don't tolerate chemotherapy for stem cell transplant well. Transplant is thus typically performed with low or no conditioning. And as a result, many of these infants have poor immune reconstitution, or it's more common for them to have poor immune reconstitution. And you can see on the right-hand side here that especially in the setting of HLA haploidentical transplant with conditioning, while their survival is only about 50%, and while it's not a lot worse than those infants with RAG, they do have more late complications. And so gene therapy for Artemis deficient SCID has been pioneered by Mort Cowan and the group at UCSF. And so they ran a lentiviral based phase 1, phase 2 trial with busulfan targeted at very low dose, and reported 2 years ago on a phase 1/2 trial treating 10 infants. They had almost 3 years of follow-up, and these infants all had immune reconstitution over the first 9 months. They had TREX that were detected quite early in development, and lymphocyte proliferation that normalized by 9 months. 5 of these patients came off IVIG, and 4 responded to vaccination. One patient required CMV-specific T cells in a second infusion, but still immune reconstituted. And so this trial is still recruiting in a phase 1 UCSF study, expansion study, targeting 25 infants. And there is also a gene therapy trial in Paris targeting 5 infants. So I think, as I said, when we think about infants or children with diseases where transplant outcomes are not predicted to be as good, it's a little easier to think about where the role for gene therapy might be. But how we find equipoise for disorders where we have good outcomes after hematopoietic transplant becomes a little harder. And so, for instance, with classical X-linked SCID, or if if patients are not infected, there's really no difference in outcomes between matched sibling donor transplants and alternative donor transplants. And newborn screening is improving those outcomes. On the gene therapy side, it's a very rapidly evolving therapy. We have safer tools than the original gamma retroviral vectors with the transition to lentiviral vectors. But these infants we have demonstrated that these infants do need some level of cytoreduction, less busulfan than what's needed for transplant, but they still need some level of cytoreduction to get full correction in all lineages and become independent of IVIG. And there are long-term risks in terms of the potential for oncogenicity that continue to be concerning. So I'm sort of highlighting here two reports on gene therapy for X-linked SCID. The first is one that was run sort of in parallel in Boston, in Europe, and in a number of centers in the US. And this was a retroviral vector, but the safer form of the retroviral vector with SYN inactivation of the enhancer. So less strong promotion of the expression of the gene. And 9 boys were treated. 7 of them reconstituted with T cells. And the integration sites were less concerning. While they are still integrations in these proto-oncogene sites, they're not preferentially in those sites. And though this trial did not include busulfan, and there was no multilineage reconstitution, meaning these boys only got reconstitution of their T cells, not of B cells, and we weren't able to show any gene marking in the neutrophils or the other cells. Alternatively, the trial run out of St. Jude was published a few years ago. This trial is a lentiviral-based vector trial. They published on the first 8 patients, and they had multilineage vector marking. So they got engraftment of hematopoietic stem cells with low dose of busulfan. They initially had no clonal dominance by integration site analysis, although there's recently been the report of some clonal dominance, although no concern for AML or leukemia of any sort. These infants immune reconstituted by 3 to 4 months after therapy. One required a second infusion, and their previous infections cleared. They normalized their IgM levels quickly. 4 of them were off IVIG by the time of this report, and 3 had responded to vaccine. This trial, however, is now on temporary hold because of what I mentioned about the finding of some clonal dominance. And so right now, there are several open recruiting clinical trials in gene therapy for X-linked SCID, including 2 in China. The Boston trial is open, and this is a multi-site trial. It includes busulfan conditioning, targeting slightly under where the lowest dose that's used in transplant for SCID is targeted. And then NIH and St. Jude have the companion trial to the one I just presented that is open for older children. So for those who are 2 years of age and older, including children who have had a failure of a prior hematopoietic transplant. And so while there are increasing numbers of curative options for patients without matched sibling donors, alternative donor transplant or gene therapy remain good options for these patients with X-linked SCID. So I'm going to move on and talk about some other disorders, the first being Wiskott-Aldrich. So boys with Wiskott-Aldrich can have residual or complete absence of the Wiskott-Aldrich protein. And that means that it's a very heterogeneous disease with clinical manifestations ranging from severe persistent thrombocytopenia, eczema, infections, and autoimmunity, and there are boys who have all of those, to sort of mild asymptomatic thrombocytopenia. The majority of boys suffer inflammatory complications that increase over their lifespan. and have an increased incidence of autoimmunity and remain at risk for developing lymphoproliferative disorders and lymphoid malignancies. And so increasingly, people really think that without definitive treatment, these boys are less likely to survive beyond their second decade. And so curative therapy is appropriate to consider, even for those who are less symptomatic early on. And this is, again, like SCID, a place where alternative donor transplant is associated with quite good outcomes, and there's no statistical difference by donor type in the outcomes that we see, including relatively recent cohorts of boys transplanted for Wiskott-Aldrich syndrome. But there is one component that remains concerning, and that's the issue of mixed chimerism. So in mixed chimerism after hematopoietic transplant, we see an increased incidence of autoimmunity and, sorry for the typo, thrombocytopenia. And this was demonstrated very nicely in a fairly historic cohort of patients transplanted for Wiskott, where they looked at the level of myeloid chimerism and compared it to the platelet count post-transplant. And you can see that those boys who have low level of donor hematopoietic stem cells, while they have reversal of the Wiskott-Aldrich T phenotype and their T cells work well and they respond to vaccines, they do not reconstitute platelet counts at a normal level, and so they remain thrombocytopenic. And this is informative both in terms of potential options for gene therapy, but also in terms of where we need to aim for in terms of correction in the setting of gene therapy. And so there have been several studies of gene addition therapy. This was a study, again, with the same lentiviral vector conducted in multiple centers. It is a lentivirus-based vector that has the full Wiskott-Aldrich gene and a small part of the promoter. So you get gene expression, but not super high level gene expression, as a way to sort of try to protect against oncogenic events. These boys got busulfan and fludarabine conditioning, and on interim analysis, all patients are alive and well. They have sustained multilineage gene marking, and there's universally improvement in eczema, infections, and bleeding. But those boys with lower levels of Wiskott protein expression have poorer platelet count reconstitution, and there are two boys who had less robust immune recovery. And when we looked specifically at the boys transplanted here in Boston, what we were able to demonstrate was that those boys who had gene therapy with a higher number of edited genes in each cell had better platelet recovery, and those who had lower vector copy numbers in each cell had poorer platelet recovery. And similarly, those boys who reconstituted their regulatory T and B cells did better in terms of being clear of autoimmunity post-transplant. And so the Italian sponsor TGED is planning to open a trial using this vector, now renamed OTL-103, for treatment of boys with severe Wiskott-Aldrich syndrome. Although the trial is not open yet, it does have an NCT number, and I'm sorry I didn't include it here, but I can give it to you in the chat later. And so, gene addition sort of is like a spray. You're spraying the corrected gene into the cell, but not really directing it where the abnormal gene is. And so gene editing is a more elegant approach in that you're trying to give a site-specific modification. And so CRISPR is sort of been a revolutionary part of gene editing. It requires two components. It requires a nuclease capable of cutting the DNA, and then something to guide the specificity. So you want the DNA to be cut in a very specific place. And what happens after that cut is you can either have disrupted the gene, so you stop expression of an abnormal gene, or you can try to repair the gene at that site where the cut happened. And so, as I said, the goal can either be to disrupt a gene so that an abnormal gene can't function, or correct a mutation. There are two ways. So when CRISPR-Cas goes into a cell, this complex creates a double-strand break. It breaks through both strands of the DNA in a very specific location. And how the cell fixes that is very important in terms of the effect of gene editing. So there are two ways. It can fix it by non-homologous end joining. So it just sort of shoves the two ends back together some way and maybe put some genetic material in the middle. And it might fix it correctly, but it might not. There's nothing to tell it how to do the fix. And then there's homology-directed repair. And in this instance, with this CRISPR-Cas complex that you put into the cell, you also put like a little template, a genetic template. And that genetic template tells the cell how to repair the break. And both of these approaches have been employed. And so with Wiskott-Aldrich, let's skip here. So with Wiskott-Aldrich, the idea has been to try to use gene editing for Wiskott-Aldrich with homology-directed repair. And so the idea, as you can see on the right with this little schematic, the idea is that the CRISPR-Cas goes in and creates a cut right at the start site of the Wiskott-Aldrich gene in the cell. And it inserts a cassette that has the normal entire Wiskott-Aldrich gene. So the cell, instead of using the dysfunctional one that's downstream, uses the correct one because that's what it sees first. And so this has not been done in clinical trials yet. But Melissa Pele and colleagues at MD Anderson did this in wild-type hematopoietic stem cells, and they were able to get integration into 50% of the cells. So that already tells you that's kind of at that level of correction that we need to be for Wiskott. And then in the T and B cells, they only got 10%, but that was enough to restore function in the T cells and B cells, as I had mentioned. So this starts to get at that idea of, can we use information from transplant and from gene addition therapy to inform how we do gene editing? And so CGD is another place where while transplant outcomes for recipients of matched sibling donor are quite good. On the right, you can see in the upper panel that those children who receive a transplant from an alternative donor have a slightly lower probability of survival. And certainly those children who have a poor performance status coming into transplant, which many children who've had multiple infections or other complications of CGD do, have a poorer overall event-free survival after transplant. And so while hematopoietic transplant can lead to durable resolution and lowers the burden of disease, there are maybe some populations of patients that we can predict will not do as well. And the PIDTC did a small retrospective study of autosomal recessive CGD and demonstrated that while overall survival was quite good at over 90%, there was a significant population of patients who had graft failure and needed second transplant. And so can we do better than that? And there are currently 2 gene addition studies ongoing of gene therapy for autosomal recessive CGD. These are, one is at the NIH and one is at Great Ormond Street using a very similar vector. And both of these are open to sort of toddler age boys and girls. And then for X-linked CGD, this is again a study led by Don Cohen. It was run simultaneously at several centers, including ours. And there were 9 boys who received lentiviral-based therapy for X-linked CGD. 2 of them died early of infections they had coming into gene therapy. 6 have had stable integration and correction with normal oxidation of their neutrophils. but one who had initial high levels of oxidative burst from neutrophils had decline of that function, and that's really consistent with that we didn't get enough stem cells transduced with the lentiviral vector. And you can see here in the yellow is the boy who had loss of function, loss of gene marking. And 4 additional patients were treated since these initial 9. And one of those 4 had sustained high level of neutrophil function, but 3 had initial high level of function and then declined. And so, this is not a very satisfying outcome for gene therapy. And the French group had a similar finding. They had just 4 patients, but 2 of them had a defect in their recovery of neutrophil function. And they were able to actually look and interrogate and see that really the issue was about how much inflammation was in these boys at the time of this gene addition therapy. And so at least they're getting to a place where they can maybe predict which boys could potentially not benefit from this type of gene addition therapy, and pushes us to ask, can we do better? And so gene editing in this setting is very appealing, right? So the idea, especially in X-linked CGD, is that there's a common mutation. And so you can use CRISPR-Cas9 to put the normal version of the gene exactly into the site that the gene should be in. And when they did that in preclinical, in the Petri dish, not in people, they were able to get repair of 20% of the hematopoietic stem cells. And again, that's right around the level we need for normal function in a person. They had functional mature myeloid and lymphoid cells, and there were no integrations that happened outside of the gene locus they were targeting. None of the integrations happened outside of the CYBB gene locus. Alternative way of gene editing that is even a little more elegant is the idea that instead of inserting a whole gene, and instead of making a double strand break that the cell may mess up how it fixes, you can target one DNA base. And so the idea here is part of your complex that you put in with the template and the Cas9 and the enzyme is an enzyme that does a specific change. So in this instance, on the right in the schematic, you see that this enzyme is changing a G to an A, or an A to a G. This is a powerful way to correct a single base mutation. And this approach may be more accurate and less risky than some of the other gene editing approaches. And the group at NIH is pursuing this for X-linked CGD. And so they have demonstrated that they can get up to 70% correction using this approach with, again, no inaccurate editing. And so they are opening 2 trials. One is for X-linked CGD and one for autosomal recessive CGD, both with a gene editing approach such as this. And these will be first in human clinical trials. And finally, just to really push towards the future, all of the therapies I've talked about so far, all of the gene modification therapies, involve taking the hematopoietic stem cells out of the person, manipulating them in the lab, and then putting them back in. But people are working on how you could target the stem cells without taking them out of somebody's body. And this would obviate the need for busulfan conditioning, for instance, because those stem cells are already engrafted in the person. And so there's been a proof of principle with XSkid in a dog model of SCID. And there were 2 puppies who were treated with, so the idea is you package the viral vector outside of the body and then inject it into the body, and the viral vector homes to the stem cells and corrects the stem cells. And so they've been able to use this type of in vivo gene therapy, using either a CRISPR-Cas setting or gene editing to correct the X-linked skid in these two puppies. And so, just finish by saying, I think it's really important to think about each patient uniquely and to think really about what and when, what type of risk is appropriate. All of these therapies come with risk, but I think especially in the rare disease setting, it will be very hard to have long-term outcomes as we're making decisions about current therapies. The therapies are changing very quickly. We don't have 10 years of data from today's gene editing to even inform what the best next approach is. And so one of the things that I think is important when I talk with families is to sort of talk about what their tolerance for unknown versus known risks are. And I think we're all a little different in terms of that. And I also think there's some people who really want to be on the forefront and want to be exploring, be the first patient to get a new therapy. And there's some people who really don't want to be the first person getting a new therapy. And I think that has to be part of our decision making, because we're in a place where we have a lot of choices and a lot of options for our patients. I think those decisions are different when we start talking about more common diseases such as sickle cell disease and things like that. And I think it's really important to keep the discussion in the setting of rare disease. And I guess it's appropriate that primary immune deficiencies are rare diseases, and we're right getting upon Rare Disease Day and Rare Disease Month. But I do think that the decisions and the thought around these choices need to be a little bit different in this setting. And these are my acknowledgments. This is a team sport, cannot do it alone. And thank you so much for inviting me, and I'm happy to take any questions.
Wonderful. Well, thank you so much, Dr. Prockop. Give me just a minute while we get set and ready to go for the Q&A. All right. So we've got a couple of great questions to get us started, but if you think of anything throughout, folks, we definitely encourage you to put your questions in the Q&A box, and we'll go ahead and we'll chat through them. And again, the ground rules for Q&A are being shared in the chat, so just please take a look at those before you enter your question. All right, Dr. Prockop, our first question. Do you see a future of gene therapy being a treatment option for those with antibody deficiencies or CVID? Or is gene therapy likely to remain most beneficial for conditions with specific gene targets?
So, so I think there are different forms of gene therapy. When people talk globally about gene therapy, for instance, some people, when they're talking about gene therapy, are talking about genetically modified CAR T cells. And those, for instance, are being used outside of the setting of specific gene disorders, and in the setting, for instance, of lupus now. And so, I see a world where some of these genetic therapies will be applicable even when you're not doing a replacement of a defined gene. And it may be that we figure out that even if we don't know what gene is causing somebody to not make specific antibodies, that we can augment some of their other processes with gene therapies to make them better. I think that's a future reaching version, but I wouldn't say that it's out of the realm of possibility.
Thank you. I know we have a lot of folks with CVID in our audience, so I think that is giving folks a lot to think about for the future. So thank you. All right, next question. As we learn more about the safety and long-term outcomes for gene therapy, is there a future where gene therapy is an option for adults who were maybe treated with stem cell transplant when they were children, but now they've aged, they're in their 30s, 40s, and 50s? Is gene therapy an option for those folks?
Yes, I absolutely think it is. And if, in fact, one of the ex-SCID gene therapy trials now is open up to people, I think at the age of 50, and includes being open for people for second transplant. So, and the CGD gene therapy trials additionally are rolling out specifically for patients over the age of 18. And I think it makes a lot of sense to think about those patients who, because of multiple infections or accumulated organ dysfunction, may become not good candidates for transplant, but are still good candidates for gene therapy. So I absolutely think that makes a lot of sense.
Wonderful. All right, next question. So I want to go back to a little bit earlier in your talk when you were talking about ADA SCID and gene therapy for ADA SCID. This individual wanted to know, did they understand correctly that the one ADA patient with acute lymphoblastic leukemia received the Y retroviral-based gene therapy?
Yes, that's absolutely true. And the gene therapy that's being performed in the US is lentiviral based. You know, ADA deficient skin was the one place where, even with retroviral based gene therapy, there had not been a case of leukemia. And so, people frequently talk about whether ADA is different because of the way in which the enzyme rescues cells that haven't been transduced, or whether there are other variabilities about the specifics of ADA deficient SCID that made those patients less at risk for leukemia when patients with Wiscott and ex-SCID who got gamma chain based gene therapy developed leukemia. But she's absolutely right. It is the one case, and it is gamma retroviral based. And so, you know, we in the US think that the lentiviral based therapy is the way to go.
Thank you. All right, our next question. This individual shares that they've got IgG3 subclass deficiency and they suffer from frequent infections. They said, I apologize if you mentioned this earlier, but could you share more about how far away we are from gene editing and gene therapy for patients like me? Could that be possible?
Um, I think it could be possible. I I think where we are now, so the first step of stem cell based gene therapy to correct um, a monogenic disorder is to understand that gene, to understand how it's regulated. And I don't think we're there quite yet for IgG3 subclass deficiency, but I think that is really that will make it most elegant to solve that type of problem with gene therapy. But
Thank
I think
you.
we'll get there.
All right, this next individual asks, if someone has a gene that is associated with telomere shortening syndrome, is there anything currently that can be done to treat it? And they say, thank you for a great presentation.
Um, yes, absolutely. So yes, there are transplant approaches for telomere biology disorders. And those approaches are very specific to the sort of increased fragility of the cells in those patients. And so there are very specific transplant programs and transplant approaches for telomere biology disorders. And if you want to send me their name or contact, or have them contact me, I can put them in touch with somebody. And people are working on gene therapy for some of the telomere biology disorders as well.
That is great to hear. Thank you. All right, this next individual asks, what happens when children with edited genes have children? Did their offspring inherit the edited genes or the original genes prior to altering? I find that such an interesting question.
It's a great question. And with the approaches we're doing now, their children would not inherit the edited genes. So they would inherit the uncorrected prior genes. Um, but it's a great question.
That is fascinating. All right, next question. Are there any current or planned gene therapy studies for other immune deficiencies for kids that depend on IG therapy? This particular person was asking about, I think it's the TACI gene mutations, TACI,
Yep.
that cause PID.
Yeah, and right now there are not. Um, but I there there isn't a reason why not. Except that right now I think there's still concern that the potential for toxicity um of gene therapy and the need for busulfan conditioning to get the fixed stem cells into the bone marrow, it still is associated with toxicity that's higher than the toxicity of IVIG and maybe prophylactic antibiotics and things like that. I think as things get um, less toxic, as we understand better what the toxicities associated with each component of the therapy are, and as we get away from needing busulfan, which I think we will, then I think it opens up a lot of those doors.
Thank you. All right, this is a great question as well. So we know there are a lot of comorbidities that are experienced by people with PID. How does gene editing work with comorbidities? For example, if they have immune deficiency and another genetic abnormality, how would that work?
Yeah, so it's a great question. Right now, the immediate toxicity of gene editing, gene therapy is the need for at least low dose busulfan to essentially what that's doing is making room in the bone marrow. So it's sort of killing off some of the dysfunctional but patient's own stem cells so that the new fixed stem cells can find a niche in the bone marrow to settle in and engraft in. Now, the dose of busulfan that's used for these is quite low, and so it's at a level where people who, for instance, have already had infections in the lung and stuff like that can probably tolerate. Some of these regimens are requiring higher levels of busulfan or more chemotherapy. So it's really about, can the person tolerate the chemotherapy? Um, the edited cells themselves, I thus far have not had any toxicity associated with them. And so much of the field is trying to think about how to get these cells to engraft, to go to the marrow and engraft and stay there forever without chemotherapy. And there's some approaches that are being used with antibodies. And if in vivo gene therapy works, that could be another way that we avoid the need for chemotherapy. And so I think that is a place where gene addition therapy and gene editing may have an advantage over historical transplant approaches in terms of having a lower toxicity profile with the conditioning.
Thank you. All right. Kind of a spin-off on that. This individual asks, in comparing stem cell transplant versus gene therapy for WAS specifically, while there's excellent survival in both groups, even with multiple donors, do you feel that with such excellent survivals, we can begin looking at quality of life and higher event-free survival comparisons between the two groups in making treatment decisions, and coming to an ultimate uh, uh, decision on which is sort of the better fit for WAS in particular.
Yes, I absolutely do. And I think not just event-free survival and overall survival and quality of life, but I think it's really important to look at the burden of things like graft versus host disease, and poor graft function that the first requiring prolonged steroids and other therapies, and the second requiring sometimes a second transplant. I think it's absolutely important to come up with ways to sort of measure the burden of those therapies, in addition to quality of life measures. That's exactly where I think we need to go.
Thank you so much. All right, going back to the question about what age you can get gene therapy and can older patients receive it after they've gotten a stem cell transplant. This person just wanted to confirm, is gene therapy less beneficial the older the patient is? And they're asking specifically around X-linked SCID.
Not not necessarily. I don't think we don't have even any preliminary data from that first trial in X-linked SCID that allows older patients and patients who've had a prior stem cell transplant. So I think it's very hard to, without any data, it's hard to know if there's an age beyond which it gets less likely to work, or an age where it works better, or other things that inform how well it works or doesn't work. I think it is the tolerability of the approach is something that makes it possible to explore in that setting. Whereas I think second transplant can be pretty toxic for people who have had a first transplant or SCID and now have poor immune function. There are also other approaches for that idea of poor immune function after a first transplant for SCID that are non-gene therapy approaches and non-second sort of second transplant approaches with just antibody-mediated conditioning that are also being explored. So I think it's an active area of exploration. Here, our plans for our next gene therapy trial for SCID absolutely include second transplant and transplant for older patients with inadequate engraftment or waning immune system after a first transplant. So yes.
Thank you. All right, so we have a number of folks also asking about other diagnoses that are not currently supported by gene therapy or currently, in clinical trials for gene therapy. And these folks are just wondering if, in addition to CVID and other antibody disorders, in the future, is there a possibility that we might see gene therapy for hypogam or Good syndrome or APS type 1 or XLA?
Yes. I think the caveat for all of those is if you have a disorder where so I should back up. So far, none of the gene therapies is able to get correction at higher than at most about 50% of the stem cells. And so if you have a disorder where there's no intrinsic advantage to a cell of having the normal gene, and you need high levels of correction, we don't have that technology yet. If without a high amount of chemotherapy beforehand. So the only place where that's been able to be achieved is in the setting of metabolic disorders. But for the immune deficiencies, if you have a disorder where you need very high level of correction, that's been a challenge in the immune deficiencies. But I think barring that, all of those disorders that you just mentioned would be potentially amenable to gene therapy. And I think the idea of gene editing and the CRISPR-Cas approach is that people don't have to spend years developing a vector. They just sort of developed a little piece of DNA that tells the whole complex where to go. And so that will significantly bring the development costs down it will significantly speed the time from sort of the lab and the mouse experiments to getting these things into clinical trials. So I think the pace at which things move is going to change remarkably.
Thank you so much. All right, next question. So we're hearing a lot about AI in the medical world, as we all know. This individual wants to know, do you believe that AI will accelerate the understanding of different gene expressions and functions and thus the potential treatment via gene editing?
That's a great question, and I would say yes. Um, I don't have an example yet, but I would say yes. Certainly. So what I would say is there's already AI involved in things like um where I showed that how the French are trying to understand which CGD patients don't get good engraftment of a gene addition therapy for CGD. That kind of analysis of huge number of data points with a not just a program that's saying which data point falls out because it's seen across all patients, but how do those actually connect, and are there specific pathways that are important? That's already involving AI, and so I absolutely think AI will be crucial for how we get to the next steps.
Thank you. All right, this individual wanted to go back to one of your last slides where you were talking about the in vivo gene therapy that was done on the two puppies. What was the outcome and how far out are they now?
So the report only goes to 16 months. And so at 16 months after therapy, they had excellent immune reconstitution.
Well, that is wonderful to hear. This next individual shares that they're new to the world of gene therapy and gene editing. How would you start that journey if you wanted to see if you or your child was a candidate for gene therapy or gene editing for this type of treatment? What would be your first step? Like, where would you go? Who would you talk to? All that
Uh,
good stuff.
so I think most immunologists would be able to help you navigate. I think the clinicaltrials.gov site is really helpful, but it takes a little bit of navigation in terms of like what terms it accepts best and things like that. I might start there.
Wonderful. And yes, and you shared a bunch of great trials in your talk, and we went through and shared those links in the chat for the vast majority of them. So if you were interested in learning more about any of those trials, past, current, or not even yet recruiting, definitely check out those links. They're all to clinicaltrials.gov, and that's where you'll be able to find information and next steps for if you would like to seek that for yourself or for your family member. All right. Well,
And
that was it. Oh, go ahead.
I should have plugged IDF because I think you guys are also a great source of that type of information.
Thank you. Thank you so much. All right. Well, on that, we are going to wrap up the Q&A. Thank you everyone for these really thoughtful questions. Thank you, Dr. Prokop, for wonderful, thoughtful answers. I think you gave us all a lot to think about, and we're definitely excited to see what lies on the horizon with this incredible science. And it seems like it's changing all the time, and more types of PI are becoming candidates for these types of treatments every day, it seems. So, thank you so much for providing all this wonderful information this evening and for a great Q&A.
Great. Thank you all so much. This was really fun. And like I said, I'm happy if anybody wants to reach out and has specific questions, I'm happy to take those offline.
Wonderful. Well, thank you so much again, Dr. Prokop. I know you probably want to get on with the rest of your evening, so I have a few more slides to wrap up with our audience, but we'll definitely let you go. And thank you so much again. We really appreciate your time tonight. All right, everybody, just a couple more slides to get through before we wrap up. And I just want to thank our audience members again for your participation and engagement in the Q&A. Those were really some great questions, and we really appreciate you taking the time to ask them. So thank you so much. So now I'm just going to quickly share about a few of IDF's resources and upcoming events. So as you all probably know, primaryimmune.org is your go-to website for additional resources, upcoming events, and more. All of our materials are free to access, print, or mail directly to you. If we didn't get to your question tonight, we encourage you to contact our board-certified patient navigator through our Ask IDF program. She will personally connect with you to tackle your question and to direct you to appropriate resources. You can even take IDF on the road with our engaging podcast series. You can find programs like Bold Conversations, Undiagnosed, and Chronic 20s by searching for the IDF podcasts wherever you find your podcasts. And we also have a YouTube channel where you can find recordings from all of our digital education events, and tonight's program will be available in the next couple of weeks. Applications are now being accepted for IDF's 2025 Research Grant Program. This program supports research initiatives focused on improving patient outcomes and expediting the time to diagnosis for individuals affected by PI. To submit your project for consideration, download the application, which we have linked in the chat. And just wanted to share an important reminder that open enrollment for Medicare is happening right now. If you are currently on Medicare Advantage and the coverage does not provide you with affordable access to medications and medical services, you have a window of opportunity to change to Original Medicare. The open enrollment period to drop your Medicare Advantage plan and enroll in Original Medicare is from January 1st to March 31st. Contact Medicare to make a change to your plan. Are you looking for ways to connect with others who are also navigating life with a PI? You might be interested in joining one of our many Get Connected groups. These groups are free, virtual, volunteer-led opportunities to connect with others with PI all over the US. We also have groups for parents, caregivers, partners, etc. So you don't even have to personally be impacted by PI yourself. We'll have a group for you. We offer location-based groups, so groups in your city or nationwide groups, and everything is on Zoom, so you can really join any group, time, leader, etc. that works for you. We also have facilitated support groups for young adults, parents and caregivers, and spouses and partners. IDF is offering several virtual screenings of our new documentary, Compromised: Life Without Immunity, via Facebook Live. Visit the link in the chat for more information. And IDF is so proud to offer educational programming each month, and we also host different events throughout the year. Coming up next, we have a Lunch and Learn in March discussing post-transplant outcomes for CGD, and we have 2 volunteer orientations coming up in March and April. Additionally, we are thrilled that registration is now open for this summer's Teen and Tween Escape in Cincinnati, Ohio. And if you have kids living with PI who are ages 10 to 18, we encourage you to register for this exciting event. And siblings within that same age range are also welcome to attend. All right, before we wrap up, I'm just going to quickly launch our poll, our post-test poll, so you all can tell us what you learned this evening. And again, you'll have 30 seconds to answer. And while everyone's taken a moment to answer the poll, I also want to encourage everyone to take the after event survey and let us know how you like tonight's program. All right, and as we conclude, I again want to thank our incredible sponsors for supporting our education initiatives all throughout the year, and Dr. Prockop for sharing her time with us this evening and providing this crucial information for our community. We appreciate everyone joining us this evening, and we hope to see you back for future programming. I'm going to leave the webinar up for just a few moments in case anyone wants to revisit all those links that we shared. And otherwise, we thank you so much for joining us tonight, and we hope you take care and have a wonderful evening. Thank you so much.